Welcome to Chapter 30, Anatomy of the Heart. In this lesson, we explore the heart, our body's central pump that sustains every cell with oxygen and nutrients. The heart is a remarkable muscular organ that contracts tirelessly throughout life, maintaining the circulation of blood through both the pulmonary and systemic circuits. By the end of this chapter, you'll understand how its anatomy supports its essential function, how the chambers, valves, and vessels work in harmony to keep the body in a state of dynamic equilibrium. This image introduces the internal anatomy of the heart through a real dissection specimen. Prominently displayed are the chordae tendineae, the thin, tendinous cords that anchor each cusp of the atrioventricular valves to the papillary muscles within the ventricles. These structures play a vital mechanical role, preventing valve prolapse during ventricular contraction and ensuring that blood flows in a single direction. Observing the heart in this dissected form allows us to appreciate its three-dimensional architecture, the thickness of the ventricular walls, the position of the septum, and the elegant engineering that enables constant circulation. This physical representation bridges textbook diagrams and clinical reality, grounding our study of cardiac anatomy in the tangible structure of the heart itself. The heart is located within the thoracic cavity, nestled between the lungs in a region known as the mediastinum. It lies just behind the sternum and slightly to the left of the midline, with its apex pointing downward toward the left hip. This positioning allows the heart to be protected by the rib cage while remaining close to the major vessels that enter and exit its chambers. Understanding this orientation is key for interpreting clinical procedures such as auscultation and cardiac imaging. At first glance, the external anatomy of the human heart reveals four chambers, two atria located superiorly and two ventricles located inferiorly. The right side of the heart handles deoxygenated blood returning from systemic tissues, while the left side pumps oxygenated blood received from the lungs to the rest of the body. Distinct surface markings, such as the coronary sulcus and the anterior and posterior interventricular sulci, house important coronary arteries and veins that nourish the myocardium. The heart's orientation, grooves, and visible vessels are all anatomical clues to the pathways of blood flow and energy supply within this organ. From a posterior view, we see how the great vessels attach to the heart and how the atria serve as receiving chambers. The superior and inferior venae cavae channel deoxygenated blood into the right atrium, while the pulmonary veins deliver oxygen-rich blood to the left atrium. This view also highlights the coronary sinus, a large venous structure that collects blood from the myocardium itself. Observing the posterior surface gives us a clearer understanding of the heart's venous return system and how blood flow completes its circuit through both pulmonary and systemic loops. Looking again from the anterior side, the heart presents a more dynamic structure. The right ventricle dominates the anterior surface, while the left ventricle forms the heart's apex. The auricles of the atria are visible as ear-shaped extensions that slightly increase the chamber's capacity. The pulmonary trunk emerges from the right ventricle, dividing into right and left pulmonary arteries that lead to the lungs, while the ascending aorta curves upward to distribute oxygenated blood to the body. Together, these features demonstrate the elegant arrangement that supports unidirectional blood flow and coordinated cardiac function. To maintain this one-way flow of blood, the heart is equipped with four valves, two atrioventricular and two semilunar. The right atrioventricular, or tricuspid valve, prevents backflow from the right ventricle to the right atrium, while the left atrioventricular, or mitral valve, serves the same function on the left side. The pulmonary and aortic semilunar valves ensure that blood ejected into the arteries does not return to the ventricles during relaxation. Each valve opens and closes in response to pressure differences, providing the rhythmic flow essential to cardiac efficiency and health. The tricuspid valve, located between the right atrium and right ventricle, consists of three flexible cusps anchored by chordae tendineae to papillary muscles. When the right ventricle contracts, pressure forces the valve shut, preventing blood from regurgitating backward. The coordination between the chordae tendineae and papillary muscles ensures the valve's proper closure during systole. Dysfunction in this valve, known as tricuspid regurgitation, can compromise venous return and lead to systemic congestion, a reminder of the valve's critical mechanical role in maintaining cardiac efficiency. The pulmonary semilunar valve is situated at the junction of the right ventricle and the pulmonary trunk. It consists of three crescent-shaped cusps that open as the ventricle contracts, allowing blood to enter the pulmonary circuit. When the ventricle relaxes, blood pressure within the pulmonary trunk causes the cusps to fill and close tightly, preventing backflow. This passive but precise action ensures that deoxygenated blood moves only forward, toward the lungs for gas exchange. Valve integrity here is essential for preventing pulmonary hypertension and maintaining low resistance flow through the pulmonary arteries. On the left side of the heart lies the mitral, or bicuspid, valve between the left atrium and left ventricle. Its two strong cusps, supported by chordae tendineae, prevent the backflow of blood during ventricular contraction. Because the left ventricle generates the highest pressures in the heart, the mitral valve must withstand significant mechanical stress. Damage or calcification of this valve can lead to mitral stenosis or regurgitation, conditions that disrupt the efficient delivery of oxygenated blood to the systemic circulation. The aortic semilunar valve guards the entrance to the aorta and operates in tandem with the left ventricle. During systole, it opens to allow oxygenated blood to flow into the aorta. During diastole, the valve closes tightly, preventing blood from returning to the ventricle. Its structure is remarkably strong and durable, with each cusp precisely shaped to handle the intense pressure of systemic circulation. Any dysfunction here, such as aortic stenosis, can lead to reduced cardiac output and increased workload on the left ventricle. The human heart functions as a double pump, circulating blood through two distinct circuits. The pulmonary circuit carries deoxygenated blood from the right side of the heart to the lungs, where gas exchange occurs. The systemic circuit then distributes oxygenated blood from the left side of the heart to all body tissues. This separation ensures that oxygen-poor and oxygen-rich blood never mix, allowing efficient oxygen delivery and waste removal. Understanding these circuits is fundamental to grasping how the cardiovascular system maintains homeostasis and supports the body's metabolic demands. Even though the heart is responsible for pumping blood throughout the body, it requires its own dedicated blood supply through the coronary circulation. The right and left coronary arteries branch from the base of the ascending aorta, encircling the heart in the coronary sulcus. These arteries supply oxygen-rich blood to the myocardium, while cardiac veins collect deoxygenated blood and return it to the coronary sinus. Interruption of this circulation, such as in coronary artery disease, can lead to ischemia and myocardial infarction, emphasizing the vital importance of this vascular network. The left coronary artery quickly divides into two main branches, the anterior interventricular artery and the circumflex artery. The anterior interventricular artery supplies blood to the anterior walls of both ventricles and the interventricular septum, while the circumflex artery serves the left atrium and posterior portions of the left ventricle. Together, they ensure that the most muscular and energy-demanding chamber, the left ventricle, receives a steady oxygen supply to maintain its powerful contractions. The right coronary artery travels along the coronary sulcus and gives rise to the right marginal artery and the posterior interventricular artery. These vessels supply blood to the right atrium, parts of both ventricles, and the conduction system of the heart, including the sinoatrial and atrioventricular nodes. This distribution ensures that the electrical impulses governing cardiac rhythm receive a reliable oxygen supply. Blockage in this artery can result in arrhythmias and conduction abnormalities, highlighting its importance in maintaining cardiac coordination. At the microscopic level, cardiac muscle reveals its unique structural features. The fibers are short, branched, and interconnected by specialized junctions known as intercalated discs. These discs contain desmosomes that anchor cells together and gap junctions that allow electrical impulses to spread rapidly across the myocardium. This arrangement enables the heart to contract as a functional syncytium, a coordinated unit. The striations visible under the microscope reflect the organized arrangement of actin and myosin, the contractile proteins that power each heartbeat. A closer look at cardiac muscle shows the distinctive branching pattern and central nuclei characteristic of cardiomyocytes. The intercalated discs appear as dark lines between cells, marking the points of mechanical and electrical connection. This structural unity allows for synchronized contraction, ensuring that each chamber of the heart contracts efficiently to propel blood forward. The resilience and endurance of cardiac muscle are unmatched. It is capable of contracting continuously for a lifetime without fatigue, sustained by abundant mitochondria and an extensive blood supply. Zooming even closer, the intercalated discs stand out as critical features that unify cardiac muscle cells both mechanically and electrically. Gap junctions within these discs permit ion flow between cells, ensuring that depolarization waves travel seamlessly across the myocardium. This enables the heart to contract in perfect rhythm, a necessity for effective pumping. The discs also anchor the cytoskeleton, preserving the structural integrity of the tissue during the constant mechanical stress of contraction. Table 30.1 provides a summary of the coronary arteries and veins. The left coronary artery and its branches deliver oxygenated blood to the left atrium and ventricle, while the right coronary artery serves the right side of the heart. Venous return is managed by the great, middle, and small cardiac veins, all of which drain into the coronary sinus. Understanding this network is essential for interpreting angiograms and recognizing regions at risk during a myocardial infarction. In this section of the table, the distribution patterns of the coronary arteries are detailed. The anterior interventricular artery, sometimes called the left anterior descending or LAD artery, is especially important. It supplies a large portion of the left ventricle and septum. Occlusion of this artery is often fatal, earning it the nickname the Widowmaker. Recognizing these relationships helps clinicians assess damage after coronary events and guide interventions such as bypass grafting or angioplasty. The coronary veins mirror the arterial pathways, collecting deoxygenated blood from the myocardium and channeling it toward the coronary sinus, which empties into the right atrium. The great cardiac vein runs alongside the LAD artery, the middle cardiac vein parallels the posterior interventricular artery, and the small cardiac vein drains the right margin of the heart. This complementary design ensures balanced inflow and outflow, maintaining the metabolic needs of the cardiac tissue itself. To conclude, this slide shows the right side of a dissected sheep heart, a valuable model for understanding human cardiac anatomy. By examining the internal structures, such as the right atrium, tricuspid valve, and right ventricle, we can visualize the relationships between chambers and valves in 3 dimensions. Seeing these structures firsthand helps reinforce concepts of blood flow, valve function, and chamber organization. This lab experience bridges the gap between textbook diagrams and real anatomical understanding, preparing you for practical applications in clinical or research settings. In this review figure, we observe the microscopic structure of cardiac muscle. Notice the striations running across the fibers, which resemble skeletal muscle but with distinct differences. Each cell, or cardiomyocyte, typically contains a single, centrally located nucleus. The dark lines running perpendicular to the fibers are intercalated discs, specialized junctions that anchor the cells together and allow electrical impulses to pass rapidly from one cell to the next. This unique arrangement enables the heart to function as a synchronized unit, contracting rhythmically and continuously without fatigue. The visible striations reflect the organized alignment of actin and myosin filaments, illustrating how structure directly supports the heart's powerful and coordinated contractions.